Aloha. Today my presentation will be focused on the citric acid cycle, and more specifically on the first half of the citric acid cycle. We will go over the production of acetyl-CoA, and then I will go over the reactions of the citric acid cycle. And like I said, this presentation will only cover the first half of the cycle, or the first four of the reactions. Now, whenever we talk about metabolism or catabolism, to be more specific, we should also introduce another term known as cellular respiration. Cellular respiration includes all processes that are involved in the oxidation of biomolecules to carbon dioxide and electrons, and then the use of these electrons for the synthesis of ATP and for the conversion of oxygen to water. That is basically cellular respiration. The process as a whole has three stages. The first stage is the production of acetyl-CoA. That stage unites all, if I can say, primary catabolic reactions for the various biomolecules. We've already discussed glycolysis. Later on we will talk about beta oxidation and the catabolism of amino acids. The common product for all of these pathways is acetyl-CoA and electrons. Now, after acetyl-CoA is generated, we enter into the second stage of cellular respiration, and that is the acetyl-CoA oxidation. During this pathway, acetyl-CoA is oxidized to carbon dioxide, and the remaining, or the reduced, or the released electrons are loaded like the electrons from the first stage on electron carriers, which are carried to the enzymes that are part of the third stage of cellular respiration, which is electron transfer and oxidative phosphorylation. During this stage, the flow of electrons is used to generate ATP from ADP and PI, and the released protons and electrons are accepted by oxygen, converting it to water. So that's, in a nutshell, in general terms, what metabolism, or specifically catabolism, or cellular respiration is. We consume food, and the end product of that consumption is ATP, which is used for any process in the body that requires ATP, biosynthesis, keeping gradients, muscle constriction, and any other process that requires ATP. In addition to that, together with food, we also breathe oxygen, and that oxygen is critical for this process because it's the final acceptor of these electrons, converting that oxygen to water. So this presentation will focus primarily on the second stage, acetyl-CoA oxidation. That stage includes basically one pathway or one process, and that is the citric acid cycle, also known as the tricarboxylic acid cycle, TCA, or the Krebs cycle, in honor of a scientist who worked on this cycle and made most of the discoveries of the TCA cycle. So we will commonly refer to this cycle as the CAC cycle, or the TCA cycle, or the Krebs cycle. All right, now before the final product of glycolysis, though, which is pyruvate, enters the citric acid cycle, we need to convert it into acetyl-CoA. Before we do that, I wanted to go over again what is the fate of pyruvate. In other words, what can pyruvate be used for? We know that in anaerobic conditions, pyruvate, especially by microorganisms, pyruvate is converted to ethanol, or in anaerobic conditions, even in our bodies, to lactate. We can use pyruvate to make glucose through gluconeogenesis. We can also use pyruvate to synthesize some amino acids, such as alanine. Or we could oxidize and degrade pyruvate to acetyl-CoA. And then that acetyl-CoA can be either further degraded in the citric acid cycle or used for biosynthetic purposes, such as fatty acid production. Either way, though, before that pyruvate enters the citric acid cycle, it needs to be decarboxylated and activated. Now, activation. We've talked about activation before, but I, again, want to focus your attention on this because it's a common principle in biochemistry. So how is now pyruvate, or what's left of it after the decarboxylation process, activated, and why? Obviously, for that molecule to be activated, that means that we're preparing it to do something. How do we activate molecules? By investing energy in them. When we invest energy, we're increasing the ability of this molecule to do work. We usually add high energy bonds to these molecules that later on will help us harvest that energy for consequent reactions. So the activation happens by investing energy, by destabilizing the molecule, by adding an excellent leaving group, such as in this case CoA. So that is how molecules are usually activated. Why do we activate it? Because we want that molecule somehow to be involved in future processes, and we'll see how that activation will pay off later. Alright, so the oxidative decarboxylation of pyruvate is an irreversible process catalyzed by pyruvate dehydrogenase complex. You will see, and we'll talk about detail, the great variety of coenzymes that are involved in this complex. The end product is carbon dioxide and acetyl-CoA. Basically, what we've activated is the remaining acetate, and by adding the CoA to the acetate, we turn it into acetyl-CoA, which is an activated form of acetate. Now, we look at the name of the molecule, acetyl-CoA, and well, at least me, when I see it, I imagine that, we have this little tiny CoA added here, because I'm looking at it, it looks small. But in reality, the coenzyme A is a fairly large molecule, especially compared to the acetate part of the whole compound. Basically, majority of it, the greatest majority of it, is a coenzyme, and then we have this little tiny acetate group added to one of the ends. That's why coenzyme A is such an excellent leaving group. It's perfectly fine the way it is, but when we add it to something, it activates it. It forms this high energy bond that has the potential to do work. Alright, so let's continue with keeping track of the carbons, ATP used or produced, and the number of electrons that are needed or produced, usually produced because this is catabolism. Alright, before we start with the reaction itself, it is important for us to go over and basically appreciate the complexity of this enzyme complex, the pyruvate dehydrogenase complex. It's actually a massive complex. I mean, we can see the pyruvate dehydrogenase complex under a cryo-electron microscope. And that you can't just really see anything under cryo-electron EM. You have to be a fairly large complex in order for you to see it. I mean, you can see here, this is about 50 nanometer in size. This is a very large complex. It has multiple copies of 3 subunits. It's okay if you don't know exactly what they do. It's just 3 subunits, E1, E2, E3. Something interesting about this enzyme is the large number of coenzymes that are part of it. 5 of them. One of them is thymine pyrophosphate, or TPP. TPP is involved in decarboxylation reactions. These are important things for you to understand and realize, because you will see these examples in the future as well. Flavin adenine nucleotides, or FAD, you know that they're involved in electron transport. Product of B2 vitamin. The first TPP's product will be B1. NAD, also involved in electron transport. Product of B3. Coenzyme A, product of vitamin B5. It forms thioesters with high energy, thioesters that have high energy of hydrolysis. And the last one is the lipoid. It's a lipid molecule which carries electrons, but also can carry acyl groups, and we'll see how that happens. So 5 different coenzymes in this enzyme complex. It is like I said, a very large complex. All right, now let's look at the various steps of the oxidative decarboxylation of pyruvate. So in step 1, pyruvate reacts with the enzyme-bound thymine pyrophosphate, TPP, and undergoes decarboxylation. So, bam, carbon dioxide is released. We have now the acyl group attached to TPP. Well, basically you can just say, okay, we're done. When we got the carbon dioxide released, we're done. Well, that's just the first step of a very lengthy process. However, the remaining steps that we're going to go over, all of them have to do with harvesting a pair of electrons. All right, so pyruvate dehydrogenase, as you can see it here, also carries out step 2. Well, first, this is the first subunit, this is the second subunit, more like a core subunit, and this is the third subunit. So the enzyme, through its second subunit, even though they're both in contact, carries the second step as well, which is the transfer of 2 electrons in the acyl group from TPP to the fully oxidized form of the lipoyl lysine group that is part of the core enzyme. And now we form an acetyl thioester and reduced lipoyl group. All right, now we're moving to step 3, where we have coenzyme A coming in and replacing the thiol group from the enzyme and binding with the remaining acyl group to form acetyl-CoA. The other product is fully reduced lipoyl lysyl group. Now, what is the difference between the fully oxidized and the fully reduced? Well, the only difference here is that the lipoyl lysyl group has a pair of electrons that are left over from the decarboxylation and transfer of pyruvate, and transfer the remaining acetate group to acetyl-CoA. Now, this acetyl-CoA is now activated for something that would follow, and we'll see what that is in a little bit. But the electron pair has to be harvested, and this lipoyl lysine cannot just go to the oxidative phosphorylation pathway and just leave it there. Something needs to deliver it. So the way this is done is described here in reaction 4, where two hydrogen atoms and electron pair are taken from the fully reduced lipoyl lysine and transferred to FAD, reducing it to FADH2, while the fully reduced lipoyl lysyl becomes now fully oxidized. Now, the interesting thing is that the FAD in this case is a prosthetic group. When I say prosthetic group, I mean that this coenzyme is permanently bound to the protein part. So FAD cannot just leave, similarly to the lipoyl lysine. So somehow it needs to load this electron pair to something else that can deliver it to the oxidative phosphorylation, and that's where NAD plus comes in. In step 5, NAD comes in, the electron pair plus a proton are transferred to NAD plus, forming NADH, and a proton is released. Okay, so what is the net product of this reaction? Carbon dioxide, acetyl-CoA, and a pair of electrons loaded on NADH. Carbon 1 of the pyruvate is released as carbon dioxide. So this is the first carbon, or I should say the first carbons released from the original glucose molecule, since one glucose molecule produces two pyruvates, and now each of those pyruvates is converted into acetyl-CoA. That means that we've released now two carbons from that original glucose molecule as carbon dioxide. Now, the remaining acetyl-CoA will enter, if it needs to be degraded, into the citric acid cycle. I wanted to give you just a brief overview so that you can have an easy reference of the reactions of the citric acid cycle. A lot of students look at the citric acid cycle as the ultimate challenge. To tell you the truth, this is not really the ultimate challenge. We're going to talk about the citric acid cycle so much, you will learn it. Whether you like it or not, you actually have to know each one of the reactions of the citric acid cycle with its intermediates and enzymes, where carbon dioxide is released, where electrons are released, where ATP is released. You need to know that, and the reason why you need to know that is because there's so much crosstalk between this cycle and many other cycles, and we're going to keep coming to this cycle, and that's why it's so important. Yes, it will take a lot of effort to remember words that don't mean much to you. It's just memorization, but just don't worry, just keep trying. If you cannot remember the first time, just keep practicing and write it down, practicing in your mind. It'll come. Alright, so the citric acid cycle takes place in the mitochondria. That's something to realize, because where does glycolysis occur? In the cytosol. This is in the mitochondria. So the first question you got to ask yourself is, well, how will pyruvate go from the cytosol to the mitochondria? Well, we're going to talk about that. But regardless, it happens in the mitochondria. Leninger, who is the first author of our textbook, worked on the citric acid cycle as well. So that's what is he known for. All right. So the first reaction of the citric acid cycle is a condensation reaction between acetyl-CoA, or the acyl group attached to the CoA, with oxaloacetate forming citrate. Now you understand why we say that the acyl group, or the acetate, was activated when it was converted to acetyl-CoA. That high energy bond, the reason why it was placed there, so that we can break it and harvest the energy into the generating of a new bond to form citrate. What is citrate? if you look at the structure of citrate, and we'll get to that point a little later, you will see that the citrate is a tricarboxylic acid, hence the name, the tricarboxylic acid cycle. All right, next reaction, citrate is converted to isocitrate, just an isomerization. Then oxidative decarboxylation of isocitrate to alpha-ketoglutarate. Carbon dioxide and a pair of electrons are released. So there are 8 reactions in this cycle. 4 of those reactions are oxidation. So it's literally a furnace in the molecular sense of the word, but this is the place where we are burning carbon to make electrons and carbon dioxide. Alright, so third reaction, oxidative carboxylation of isocitrate to alpha-ketoglutarate. The next reaction is oxidative decarboxylation of alpha-ketoglutarate to succinyl-CoA. A pair of electrons is released together with a molecule of carbon dioxide. The next, conversion of succinyl-CoA to succinate, a molecule of ATP is released. Hydrogenation of succinate to fumarate, 2 electrons are released. Then we hydrate fumarate to malate, and then malate is oxidized to oxaloacetate, another pair of electrons is released, and then oxaloacetate can begin another cycle. Alright, so what happened with the initial substrates? So what entered the cycle? Acetyl-CoA and oxaloacetate. What happened with acetyl-CoA? It got degraded to carbon dioxide. 2 carbon dioxides were released. The acetate has 2 carbons, and 2 carbon dioxides were released. And we have several electron pairs that were also released and harvested during these reactions. The other molecule that entered the cycle is oxaloacetate. What happened with the oxaloacetate? By the time we ended the cycle, oxaloacetate was regenerated. So what do you expect would be the concentration of oxaloacetate in the cell? Do you think it's going to be high or low? And if you think one or the other, why? Well, think about it. Well, my logic is we're regenerating oxaloacetate. We're not binding it and using it. We're regenerating. As long as we have a stable concentration of oxaloacetate, the citric acid cycle can continue. If one molecule of acetyl-CoA enters, that means that a molecule of oxaloacetate will enter. So as long as we have a molecule of oxaloacetate, we can just enter the cycle. There is also another reason, but we're going to learn about that reason a little later. And that is that oxaloacetate, you've seen it already in gluconeogenesis, is the first product of gluconeogenesis, if you remember, from pyruvate to oxaloacetate and then to PEP. Oxaloacetate can also enter gluconeogenesis. So for whatever reason though, oxaloacetate has a lower concentration. We're not losing it. Nobody is, the net product is 0 basically. There's no increase or decrease of oxaloacetate. So we don't need to have a massive concentration of it in the cycle. Alright, so here's the cycle. All 8 reactions. Where do we have carbon dioxide released? Electron pairs, and what are they loaded on, and GTP. So let's look at the individual reactions now. Alright, the first is citrate formation by citrate synthase. It's pretty logical, citrate synthase. It basically what it does is, with the help of water, it releases the CoA, which cycles back to pyruvate dehydrogenase, and we have citrate. And you can see 1 carboxyl group, 2 carboxyl groups, 3 carboxyl groups, a tricarboxylic acid. Here is the electron pushing and arrow designations basically showing you how we explain this condensation reaction. Notice again the histidines that are part of the active sites. I'm reminding you of that because when we talked about amino acids, we mentioned that histidine, because of its pKa being around 6, pKr being around 6, it plays a commonly is found in active sites because it can act as an acid or a base at the same time. Second reaction, isomerization of citrate to isocitrate, and the aconitase is the enzyme that catalyzes this reaction. I oftentimes, especially when I'm in class, I would ask you, from organic chemistry perspective, how likely it is for something to come in and pull a hydrogen for a molecule like this, or a hydroxyl group from a molecule like this? And the answer is, from organic chemistry perspective, doing it in a test tube, I rarely have seen reactions like that, where a hydrogen will be pulled out of the carbon like this, out of the hydrocarbon like that. I mean, that's incredibly rare. But for whatever reason, this happens inside the active site of the aconitase enzyme. Now notice this intermediate, and I want to say, which is the transition state actually, very unstable. But what is the role of the enzymes? How do enzymes work? How do enzymes make seemingly impossible reactions possible? By lowering the activation energy. Well, how is that activation energy lowered? By stabilizing the most unstable part of that reaction, the highest energy intermediate in the reaction. It stabilizes these high energy intermediates, highly unstable intermediates, and by stabilizing them, it makes it possible for the reaction to occur. So again, the enzymes stabilize the transition states which are most unstable. So if allowed for this reaction to reach an equilibrium, 10% would be isocitrate, 90% will be citrate. That's not a very favorable reaction, and that is because of this high energy intermediate. But in the cell, reaction moves towards the isocitrate. Why is that? How can you explain that? Well, if you look at it as an isolated event, it's going to be difficult to explain. But if you look at this as part of a pathway, as part of a process much bigger than just a simple reaction, you will realize that isocitrate reaction is followed by a very favorable reaction, and we'll talk about it in a second, that pulls it towards that reaction. And whatever isocitrate is made is quickly taken and used in the consequent reaction, the follow-up reaction. And for that reason, this reaction constantly goes forward towards isocitrate. Alright, next reaction is the oxidation of isocitrate to alpha-ketoglutarate. We have carbon dioxide release and electron pair, of course, and the reaction is irreversible. Here's what happens during this reaction. And I wanted to point out, when is the carbon dioxide released? I mean, could it be released right here? I mean, technically speaking, I guess it can, but it doesn't happen. The molecule is pretty stable. But it happens, first again, what's the likelihood of pulling these hydrogens out of this carbon and oxygen and loading the electron paired NAD+, but indeed that's what happens. And then when we form this intermediate that is again highly unstable, that's where we can see and appreciate the role of the coenzymes, or actually the cofactors. I'm sorry, not the coenzymes. This is a metal cofactor. The fact that we have so much electron density in this part of the molecule and now we have metal ions that are pulling that electron density towards them, makes it possible for this bond to break after collapsing of this negative charge, and then to form another transition state, and which is also unstable. But once this happens and the decarboxylation happens, then the rearrangement again of the electron density will allow for this carbon to pick up a proton from the environment, which is very easy because the environment has them, and then form the alpha-ketoglutarate. So how many carbons have been released since the beginning of the citric acid cycle per molecule of acetyl-CoA? This is the first. So this is the first of the two carbons that are part of each acetyl-CoA molecule. Okay, next reaction, oxidation of alpha-ketoglutarate to succinyl-CoA, and again we have a carbon dioxide release. This is again an irreversible reaction catalyzed by alpha-ketoglutarate dehydrogenase complex. Obviously, dehydrogenase tells you that this reaction is irreversible, and that it's redox reaction, and it happens to be also irreversible. The alpha-ketoglutarate dehydrogenase is very similar in structure and the way it catalyzes the reaction to the pyruvate dehydrogenase enzyme complex. So, how many carbons have been released so far from the starting reaction, the citric acid cycle? 2. We entered with a acetyl-CoA that has 2 carbons, and now we have 2 carbons released up to this point. Okay, so here's the first half. First 4 reactions. Acetyl-CoA enters, oxaloacetate enters, we have 1 carbon dioxide, another carbon dioxide, we have 1 pair of electrons, a second pair of electrons. Now my question to you is, but I'm not going to answer it this time. Actually, this question is going to be part of your assignment for the next class, is, were the carbons of the acetyl-CoA that entered this cycle released as carbon dioxide or not? If yes, how can you tell? If no, again, how can you tell? And if the carbon dioxide does not come from acetyl-CoA, then what is the source of these carbons that are being released? Okay, so think about it, and I'll see your answers in your assignments. So for now, I will stop at this point, and then I will continue with the remaining part of the citric acid cycle and the glyoxylate cycle in my next presentation. Thank you for your time.